Uh, we're going to start the chapter on coordination, which we've done 4 previous videos. Now, this is the video number 5, which is going to be the what is left of the chapter is the hormones in the menstrual cycle and how they operate during the menstrual cycle, birth control, venous flytrap, auxins, and gibberellins. I hope they can be completed in this video, but if not, then they will continue in the 6th video. First of all, let's try to understand the menstrual cycle in the two main phases that it has between day 1 to day 14, which is called the follicular phase, and day 14 to day 28 is called the luteal phase. So let's just make it a little simpler for ourselves to understand it, so that we're going to talk about two phases. Now, I'm I've talked about FSH and LH, and I just said FSH and LH are two very important hormones that are produced by the anterior pituitary gland. Please remember, the pituitary gland has two portions. One is the front and one is the back portion. The front portion is called the anterior and the back portion is called the posterior pituitary gland. And the posterior pituitary gland releases ADH. So please do not mix this, and it's very important that you write. If you just write pituitary gland, you're probably not going to get a mark. You have to be sure that you are going to write anterior pituitary gland. So the anterior pituitary gland produces FSH and LH. So both these hormones are produced and released by the anterior pituitary gland. And please remember, these are glycoprotein hormones. Now, if you remember from your AS level, so there would be more proteins produced at the ribosomes and glyco-Golgi. So there'll be more Golgi and more ribosomes here, which will be producing the glycoprotein hormone. Now, in the follicular phase, we have number one, the estrogen levels rising. Now you can see this is the estrogen is the red graph. So the estrogen level rises, and similarly the FSH level also is rising. It's starting to rise as the estrogen level rises and it reaches its peak. You can see here the FSH level is also rising. So the FSH is the sort of orangish yellowish graph, and it rises. So we're having two. Now this is a positive feedback. Estrogen rises, FSH rises. And then we have the third hormone which I talk about is the LH. Now LH is low, it's very low. And then suddenly around about day 10, as the estrogen rises, the LH suddenly rises and then it peaks and then it falls down immediately. So this is called the surge. Surge means a sudden increase and then a sudden decrease. And you see the peak of the surge is on day 14. The peak is on day 14. Now this is the time when ovulation takes place. And then of course the LH decreases and then it remains low for the rest of the cycle. So the LH surge is the one which you have got to remember, is that is going to happen on day 14 and which is going to result in ovulation. And then once ovulation has occurred, now if you remember from the previous video, there's the ovary, and then we had the follicle developing, and then the follicle bursting, and then coming out, and then what was left of it was called the corpus luteum. And then the corpus luteum starts to produce the hormone progesterone. So you can see the progesterone graph is 0 entire. Now at day 14 it suddenly starts to increase. So the progesterone graph increases after day 14 and then remains high, and then around about day 26 then it starts to fall. So please understand that the main 4 hormones, so estrogen and progesterone produced by the ovary, also produced by the estrogen, progesterone produced by the ovary, FSH and LH produced by the anterior pituitary gland. This is another graph which explains this very well. You can see again we have the follicular phase and the luteal phase, we have the ovulation. And another factor which you've got to understand is that the estrogen and progesterone are steroidal hormones. And because they're steroidal hormones, they can easily enter the phospholipid bilayer, steroids, cholesterol, fats. So the hydrophobic part of the cell membrane is not going to be a deterrent, because if it was a water-soluble hormone like insulin, then it would need receptors on the cell membrane. Now in this, of course, as it can enter the cell membrane, so it is not going to be needing a receptor. So no receptor will be needed for this hormone, and it will enter the cell, and then it will have receptors inside the cytoplasm or the nucleus where it will bind and will result in transcription of certain proteins to take place. Now coming to the topic of birth control, what we need to understand is that birth control is, we want to prevent pregnancies. A woman wants to have maybe one kid or two kid, or she wants to just have spacing between the children. So she wants to prevent becoming pregnant. So there is one way is that she can start taking, there is a 21, this is called the oral pill. Now the oral pill is 21 tablets in a pack, and the woman has to take one tablet every day for 21 days, and then, of course, she menstruates, and then again she starts a new pack. So 21 tablets, and she has to take one tablet daily. So, what does this do? And what does this tablet contain? You see, the biologists, when they saw this part of the graph, they came up with a very brilliant idea. They found that, as estrogen and progesterone are high in the blood during this time, but then during this time, the interesting thing is that FSH and LH is also zero. So they thought, okay, well, then what do we do is if we give a woman synthetically a tablet which contains estrogen and progesterone. So if it contains estrogen and progesterone and the woman takes a tablet every day, so it's going to be absorbed, and her blood levels of estrogen and progesterone are going to be high. So it is going to be mimicking this area of the graph. And which means that the pituitary is not going to release any FSH or LH. So if no FSH and LH, so this whole story is not going to take place. No Graafian follicle is going to develop. No Graafian follicle develops, no ovulation, no pregnancy. So this was very empowering for women, because whenever they were developed and the oral pill came into the market, then women could decide when to become pregnant, when to have a child, when not to have a child. Of course, it went both ways. It also resulted in the increase in sexually transmitted diseases, syphilis, AIDS. So basically, the concept was that if we gave women tablets, if we gave women tablets which contained estrogen and progesterone, the estrogen and progesterone would be absorbed, it would enter the blood, and the blood will carry more, because the woman had to take one tablet every day. She had to take one tablet every day. So when she took this tablet, it was absorbed, and her blood levels of estrogen and progesterone became more. So when they became more, the pituitary detected that. So the pituitary decided, okay, so estrogen, progesterone high, so no luteinizing hormone, no FSH. Well, if there is no FSH and no LH, well, then this thing is not going to happen. So no follicle developing and no ovulation. This, of course, was how pregnancy was avoided, and it used to be taken, and millions of women still take the oral pill, and it is a very successful, and it doesn't have any side effects. So lots of women are on the oral pill, and number of children They can limit the number of children they want to have, or they can have spacing between the kids. So the birth control method was actually designed after the doctors or the biologists started studying this part of the graph, and this was the luteal phase of the graph, in which they found out that when estrogen and progesterone are high, FSH and LH is low. So this is the biological basis of the oral contraceptive pill. This is another very interesting diagram to show you all the different phases of it, and how estrogen progesterone increase in the beginning. And this is the part which is the important part for you all to see, is that how menstrual cycle, this is the shedding of the lining here, and then of course the repair, and then the phase in which there is more, the lining of the uterus becomes spongy and vascular in anticipation of implantation, which may or may not occur. And then I would like you to pause the video here and have a look at this and try and understand all the different phases of it, because it's very, very self-explanatory, this diagram. Now coming to the topic of auxins and cell elongation. Auxins are plant chemicals, and there's a whole bunch of them which are responsible in different stages of the plant cycle. But here we're going to talk only about auxins and cell elongation. Now, one of the chemicals, one of the many auxins, but one of them is indole acetic acid, which is called IAA. Now, this is produced at the growing tips of shoots and roots, and this is transported either by active transport or in the phloem sap. Now, growth in plants occurs at the meristems. Now, what is the meristematic regions are the ends, the shoot tips and the root tips, and that is where all the growth occurs in plants. So please understand which is the meristematic region, and you usually get a lot of questions on that. So meristematic regions are the shoot tips and the root tips where a lot of growth takes place. Now, growth is very easily divided in plants into, I call it, three very easy stages, and I, of course, make a mnemonic to remember it, because sometimes, remembering stuff is a little difficult. So I make a mnemonic for it is DED. So DED stands for what? DED stands for number one, cell division by mitosis. So first of all, the cells are going to divide by mitosis. So we just have, say, 4 cells. And then we have these cells divide by mitosis. So now we have 8 cells. So cells have divided by mitosis, and we have 8 cells. So cell division by mitosis. And then of course we have cell elongation. Cell elongation means these 8 cells suddenly become longer cells. Now that's a little uh, weird, because that only happens in plants. So these cells become a little longer, so we call them cell elongation. And that cell elongation is because it is the absorption of water occurs. But how does that occur? Because the cell wall is very rigid. So we've got to understand how that occurs. And then of course the third stage is called cell differentiation. Now that's why I made the mnemonic DED. So this one is for division, this one is for elongation, and this one is for differentiation. So division, elongation, and differentiation. Now, auxin has a role to play in cell elongation, and I'm going to try and explain that to you here as best as I can. First of all, what you have to understand is that the auxin binds to a receptor. This is the receptor in the cell membrane, because as you know, plant cells have a cell wall and a cell membrane. So this part is the cell wall, and this part is the cell membrane. So there are receptors in the cell membrane where the auxin binds. Now this results in hydrogen ions. It's a pumped out. When I say pumped out, that means it's going to be active transport. So the hydrogen ions which were inside here in the cytoplasm are going to be pumped out and they go into the cell wall. Now this lowers the pH. So I said acidifies the cell wall. It lowers the pH in the cell wall. And this results in activating some proteins which are called expansins. These expansins cause bonds to break. So the cell wall is not that rigid anymore. It becomes less rigid. But it is facilitated further by potassium ions diffusing in. Now you can see potassium ions are diffusing in into the cell. And potassium ions are diffusing into the cell. Of course, this lowers the water potential here inside. So it lowers the water potential inside. Now when this lowers the water potential, because suddenly potassium ions are entering the cytoplasm of the cell, now what is going to happen is that water is going to enter through the aquaporins. So once again, first of all, I said auxin binds to receptor. Then I said this causes hydrogen ions to be pumped out. When the hydrogen ions are pumped out, they're pumped out where? They go into the cell wall. So this lowers the pH of the cell wall. This of course activates the expansins. The expansins breaks the bonds between the beta glucose of the cellulose cell wall, and this results in potassium ions diffusing in. So potassium ions diffuse in. When the potassium ion diffuses in, of course, this lowers the water potential and water enters through the aquaporins. And when the water enters through the aquaporins, of course, this makes the cell elongated. So this results in cell elongation. So first we had cell division, then we had cell elongation, and then we had cell differentiation. So this is how auxins work in the process of cell elongation. Now coming to the topic of gibberellins and seed germination, G for gibberellin, G for germination. We're going to talk about the barley seed. And as you can see in the barley seed, I have taken a diagram from the book. And we have three factors which are necessary for germination. An easy way to remember is a TWO. Now T stands for temperature, W for water, and O for oxygen. Please do not say air because air contains a lot of other gases as well. So temperature means an adequate temperature which is different for different seeds. And water is of course water, and oxygen is needed for the process of aerobic respiration. And water is needed for activating certain enzymes to start working. So as soon as we say number one, first point that I talk about is water uptake initiates germination. Number two, I talk about embryo. You see, there's an embryo here which synthesizes gibberellin in response to water uptake. So gibberellin is only synthesized. Otherwise, even it's a dormant state and it is lying in a dry state in a jar, there is going to be no production of gibberellin. But as soon as we soak a seed, we find that the water uptake will start to initiate the process of germination. Now the embryo synthesizes gibberellin. Now this gibberellin has to move, it moves to the alluron layer. The alluron layer is just this layer which is just under the seed coat. So alluron layer, now when the gibberellin in response to the water uptake, gibberellin moves to the alluron layer. And when it moves to the alluron layer, here in the alluron layer, you have to remember there must be cells. It results in transcription of amylase. So the cells in the alluron layer are going to produce amylase. This amylase is normally not produced, but when germination starts, it starts to produce amylase. Now this amylase then diffuses into the endosperm. So the endosperm tissue containing starch reserve, this is the area where the endosperm is present here. So the amylase moves into the endosperm, and here of course you know starch is present. So the starch is going to be converted to maltose. Maltose to glucose. The glucose is going to be respired. Glucose respired, energy released. When the energy is released, of course, it's going to result in the growth of the radicle. The radicle is going to grow down and the plumule is going to grow up. And then it's going to develop into the shoot. So please understand the role of gibberellin in seed germination. Water uptake initiates germination. Embryo synthesizes gibberellin in response to water uptake. Gibberellin diffuses into the aleuron layer. There it causes the transcription of amylase. Amylase is transferred to the endosperm. In the endosperm, the starch is converted to maltose, maltose to glucose, glucose respired, and energy released. Venus flytrap is another topic which we need to talk about in this chapter. It is a carnivorous plant. It traps and digests small insects. They get caught in it. They are attracted because they have the leaf has nectar secreting glands on the edges. So the nectar secreting glands at the edges attract the insects enters. Now this carnivorous plant is made of two lobes, and the two lobes are open, and when an insect lands on it and starts, sort of roaming around, it actually touches the three stiff sensory hairs. So the three stiff sensory hairs which each lobe has are going to sort of give a response in which the lobes are going to sort of close up on the insect. But this insect is going to be digested by glands which secrete the digestive enzymes. Now, this carnivorous plant actually traps and digests insects because it needs the amino acid, because these plants usually survive in areas where there are nitrate deficient soils. So they have to get their nitrogen through digesting these insects. They sort of like we eat proteins and we get amino acids. So this plant is also a carnivorous plant, and it gets its nitrogen supplies by digesting these insects which are trapped inside, because the two lobes are going to close up on each other and form sort of a pocket in which this insect is going to be digested. When the insect is roaming around on the Venus flytrap, it causes deflection of the sensory hair. Now, these three sensory hairs, the stiff sensory hairs, are the ones which are actually going to cause the trap to close or remain open. So, the deflection of the sensory hair activates calcium ion channels, which are found at the base of the hair. Now, the calcium ion channels open and calcium ions diffuse in. This generates a receptor potential. Now, it's very interesting is that if two of these hairs are stimulated within 20 to 35 seconds, only then the trap will close. Otherwise, it will not close. Because you see, it would be useless if it was raining and raindrops would result in closing the trap. You don't want that to happen because you want to waste a lot of energy. Now, the trapped prey triggers the hairs, forces the edges of the lobes to seal, and this forms an external stomach, like just like the stomach that we have where food is digested and there's HCl and pepsin. So the prey digestion starts. The trapped insect stimulates the entry of calcium ions into the gland cells. And now these going to be exocytosis of vesicles which contain the digestive enzymes. And the insect is going to be digested. And the plant is going to get the relevant amino acids which it needs for its growth. So the strap stays shut for a week and then it opens, and just the remains are just blown away by the wind, and the leaf reopens. Now the Venus flytrap is a very sensible carnivorous plant. It avoids closing unnecessarily. Number one, stimulation of a single hair does not trigger a closure. Like for instance, wind blows a piece of a leaf from another tree, and one of the hairs are stimulated. So that is not going to result in the closure or if there are raindrops falling, that is not going to result in the closure of the Venus flytrap. So stimulation of a single hair does not trigger a closure. Then the gap between the stiff hairs is allowing the small insects to crawl out. Because you see, small insects are not going to be much of a very beneficial for the Venus flytrap. So plants cannot waste energy by digesting a very small meal. So it is not going to close. So this is how it prevents wastage of its energy, by closing on something which is very trivial and is not going to get any benefit from it. Now coming to the topic of action potentials. Now it's very interesting to see how action potentials are different in animals, and it's a very good comparison if you've seen the previous video of it. Action potential in animals is, the mnemonic is Nike. The depolarization is because of the sodium ions diffusing in. So sodium ions diffuse in, and in repolarization, so please remember, we have depolarization and repolarization. In repolarization, we have potassium exit. But what happens in plants? What happens in plants is, the important thing is, it's not like this. It's not sodium in and potassium exit. It's chloride out. So the mnemonic for that is very easy for you all to remember. So chloride ions move out. Chloride ions move out. But repolarization is the same as in animals. So repolarization is the same, but the depolarization is different. This would be, of course, the depolarization. This is the depolarization. The depolarization is because of the chloride ions moving out. So the action potential as compared to, this is a very easy way to remember it. Here we had the mnemonic Nike, and here we have the mnemonic Coke. So for you all to remember it, how the action potential differs in animals and how it is different in plants, is a very easy method to remember it. That finishes this video on coordination and completes the chapter on coordination. Thank you.